Hello.
Hi, Serena. How are you?
Good, good.
Is your workday over finally?
It is. I kept talking with Victoria for a while.
Oh, nice. That's nice.
Yak, yak, yak.
Okay. Yeah, I
Hi,
had the
Dee.
meeting with Howard.
Hmm?
I had the onboarding meeting with Howard Bloom.
Uh, Did that go okay this time?
Well, after a while, it took a while, a lot of back and forth, and then it finally worked. So, yeah, but that's okay. And yeah, and then I chatted a little bit with Z because Z was the only one that sticked with it to get Howard onboarded.
Oh, interesting. Okay.
Everyone else left. And she said, "Oh, I'm so glad it worked." It was really nice. And we realized she doesn't even live that far away from where we are. So,
Oh, is
and
that? Yeah, I've seen her. I've talked to her in other rooms. Is that who we were talking about earlier?
Z, yeah, I think so. Yeah, she comes here.
Oh,
Oh,
yeah. I've seen her talk in other rooms.
yeah,
She's
she's
nice.
very nice.
Mm-hmm.
Yeah, shall we just start? Hi everyone. Hi
Yeah.
BA. Hi Sarah. Hi Ram. Chewy.
Hello, everyone.
Let me ping a few people in. Maybe they. Oh, It's very limited the people you can ping in now. Did you
Is
realize
it?
that? Yeah, it's
Hmm.
just very few people, and people tell me, "Oh, ping me in when you have a room." I can't do it. It's just a few people now. So yeah, it's already. That's it.
Well, I missed a lot of rooms this week, so this will be good for me to catch up too.
Oh, okay, cool. Okay, yeah, then let's just start, and then I hope I remember everything. It will be fine. Welcome everyone to the Science Society. We have a recap room this time on Monday evening, and yeah, let's go over what we learned last week. And it was a pretty exciting week. So on Monday we had Dylan Enright. He's a PhD student in microbiology, and he's in the Glassman lab. And what he does, he studies microbiome community structures and how they change related to their environment, like how they react to the environment. And he looks at soil fungus and bacterial abundance and species richness. And the study he talked about was especially about wildfires and actually mega fires. And the interesting thing was that just by chance, um, they had collected soil, like another group had collected some soil before the mega fire for another study. And then they also had another control group where they had a very similar type of soil from very close by region that wasn't affected by the mega fire. So they had what usually these studies don't have, two control groups, and then they had the soil that was affected by these mega fires. And it's the first study of this kind, basically. And they found that a few microorganisms, survived these mega fires, and they are at the stage of characterizing them, mapping them out, who survived, and they are still looking into what are their characteristics why they survived as megafires, and then also to observe how basically the megafire soil bounces back, and if they are a huge contributor, if the diversity comes back. That is all ongoing work, but until now, just having this comparison of the same region basically before megafire, after megafire, close by region that wasn't affected, and then compare how the diversity shrinks and the few that survive, and what they will do next, basically, if they will help out this region and the soil to bounce back. So I think it's really important work, not just the microbiome in our guts, but basically in the soil is important for us, because without fungus, plants can really grow. They need the symbiosis, and we need also bacteria and all these microorganisms to basically put back the ingredients for growth into the soil. And yeah, so they are very, very important. And yeah, this is a study that finally addresses this. I wasn't aware that we know so little about the microbiome of the soil in different
Yeah,
regions.
this was a really cool tie. I made this one, and I didn't have a lot of questions for him because it was all ecology. I keep wanting to. Okay, well, if you find the fire-resistant fungi, I mean, let's figure out what the genes are. And I kept thinking of, well, do we want to introduce? And it's like, no, you can't go do that. So I ended up not having very many questions, but I thought it was really fascinating work to just because it was such a controlled study. They I think it was the speaker's advisor who happened to example, and so when he came into the lab, they already had the data, and then the fire had come through, and so they had a really it was a fortunate outcome to a really unfortunate catastrophe, but it did reveal where the fire resistant fungi are and what their species is, and they got to study how it comes back. So it was a really cool study.
Yep, I agree. And I'm looking forward to learning, what they learn from why these specifically survive and what they will do. And yeah. Okay, so next we had Dr. Manu Guri and Dr. Risi. Um, they joined us from Aalto University, which is in Sweden, and they had really cool, really interesting work. So what they did, they engineered hydrogels. So they DNA engineer hydrogels, which are light adaptive and will have different responses, and the responses reflect in different colors. And so basically, they are basically copying nature. Chameleons, they especially looked at chameleons and the adaptivity of their color to surroundings or their mood and so on. And then basically wanted to recreate that with their hydrogels. Look at the presentation. It's a really well made presentation. So they use light as stimulus. They have this material they use. These hydrogels are very, very stretchy. So you can stretch them in all directions really, really well. And they are active plasmonic materials, and you can program to have a specific color response based on the input they get. And they programmed it based on different light and heat inputs. Yeah, so if you want to look at slide number 4, it actually shows really well how they fabricated them, how they put those DNA components on, and then embed that in this hydrogel. And you can program them in any way you want. You don't have to stick to what they did. So they used gold particles to basically, with the light stimulus, to have different types of colors as an output. And yeah, this the photothermal energy creates these oscillations, and then you have basically this different outputs of colors, which are really, really cool. Yeah, it's just a really cool technique that maybe will be used in the future for all kinds of indicators. As I said, you don't have to use necessarily light or heat. You can also use sound, they said, or electromagnetic fields. So you could have them as sensors, as wearables, that basically give like a color feedback. And yeah, it's just so cool. The applications are out there. This is just a very basic research of showing that you can generate this very stretchable hydrogels that change in color. And yeah, it's up for grabs for people to invent, whatever they want to do out of it. And they are working on, making now, and they really love the sound one that kind of Jamie asked about. So they are really thinking of creating a sound responsive one.
Oh, wow, that's cool. See, I love these novel material talks. I missed this one, but it looks cool looking over the slides. So the DNA wasn't really doing anything other than providing material property. Is that right? No.
Yeah, yeah, exactly. So depending on how many C's and T's, and so you have a different melting temperature, and this then kind of with different temperatures creates then a different color spectrum
Mm-hmm.
because of the melting. And then they also have eye motifs in there in this setup, so it's also pH dependent, but they can also make DNA structures that are not necessarily so pH dependent.
That's really cool.
Hmm.
What were their killer applications, or it was just really cool?
Yeah, it's just really cool. Like we then discussed what we could do about it, and never like if this room would have never ended. I kind of had to go back to work, and we went over 2 hours, and we could have like people, we came up with more and more ways what we want to do with it.
Oh, materials for ball gowns, huh?
Yeah, like sensors for, I don't know if I ask how sensitive they are to electromagnetic waves, if they are sensitive enough to be like a visual, EEG type, that would be cool.
Oh, yeah. But this was the talk with Jamie wanting his tactile material.
Yeah, exactly. That's how he came up with that.
Oh, that would be cool. That's a good. Okay, so it was just these hydrogels with the DNA modifications, and I guess you can there's a whole creativity that could go into what coding sequence you want to apply.
Exactly. Yeah, you can change the properties really. Yeah, quite quite.
Very
It's
cool.
very cool. Yeah. Okay, let me get to the next thing.
There were a lot of talks during a lot of European talks today or this week, right? Because I missed so many rooms.
Yeah, they were. It was just by chance, but you're
Mhm.
right. Okay, so we had the microbiome room, which was pretty interesting. he let me put it up. No, mm-hmm, that's the paper. Okay, yeah. So um yeah, this room was then the postdoc was actually here, and Dr. Reish, and it's from James Collins' lab and at MIT, and they basically looked at first they started looking at what happens to the microbiome with when you use antibiotics, and what can you do to prevent basically the diversity loss of the microbiome with antibiotic treatment. And they chose antibiotic treatment because that's the most common insult to the microbiome in humans. So yeah, this paper, they looked at inflammatory disease because of metabolic changes of like a poor microbiome, and they then engineered the strain of Lactococcus lactis, and so that it would basically, after broad spectrum antibiotic exposure, how this engineered bacterium would basically resist to this strong antibiotic exposure and basically survive in the microbiome. And yeah, they were quite successful. They prevented the loss of colonization of and in they are still continuing to engineer live biotherapeutics that can also safely degrade antibiotics in the gut, in order for basically the gut to be to survive without having this major inflammation and major insult to the gut due to antibiotic treatment. Yeah, and they're also doing clinical trials um, soon, and yeah, I think it's really important. So basically, he said that we have to kind of, when we put sunscreen on. I got the phone call. When we are exposed to sunlight, we put sunscreen on, and that's really important in the future that we realize we not just have to protect our skin from major insults. We also have to start protecting our microbiome because it's really important for future health that we have a healthy one. So I think it was really cool applied science, basically.
So I came in late on this one, and I didn't get a chance to. I did because I didn't hear the talk. I didn't know if they already covered it. But did he have a position on probiotic supplements in the diet?
Well, he said, yeah, I asked him that. He said he's not a medical doctor, and there is like some sort of um, there's some people claim, you don't get the diversity back if you kind of take this very strong micro like this probiotics, but he says that it is for now. He again, he's not a medical doctor, he doesn't advise on treatments, but that's better than nothing, and also pre and probiotics are really
Mm-hmm.
important. And so far, but we have to do more work to kind of look into the diversity and what's ideally in the future. We know what your own microbiome, healthy microbiome, looks like, because it will vary from person to person, or at least family to family maybe, and you pass it on, from birth. A big part is passed on. So there has to be more work done to identify, what's your healthy microbiome and how to get it back, basically. That
Well,
would be
yeah,
the
because even if you like, okay, here is your microbiome. Well, what do you do with that? I mean, and is it healthy? Could it be better? I guess we just don't know, right?
Yeah, he says the more diverse, usually. So for now, the data looks like the more diverse your microbiome, the healthier you are and will probably be. So it's not really exactly which strains exactly you have and the amount. It's more how many of the gene pool basically do you cover, and the more the better.
Hmm. Because it seems like there could be some bad ones in a really diverse set. I guess they just get weeded out somehow.
Yeah, and we had a guest speaker actually that showed that bad ones can really produce this signals also from neurons to induce anxiety and stress and things like that. So they're definitely also bad ones. But if you have a high diversity, um, apparently it's
Hmm.
basically a healthy one. But yeah, I think there's still a lot of contradictory publications out there, and we have to learn more. Do you remember the talk we had? I think it was I forgot her name. Right now it's not coming to me. We had the researcher here that found that some microbiome bacteria produce the stress hormone, basically, that makes
Oh
us
yeah, yeah. I can't remember her name either, but yeah, there could be some nasty critters out there.
Mm, exactly. And then I think the diet and so on, probably. But yeah, so basically what they are doing is screening, more screening, and then engineer them in a way that um, they would survive and degrade the antibiotics in the gut, and that way basically make some strains survive. So
Hmm. And he's already in clinical trials.
mm, yeah, they are yeah, they are switching also to a clinic. Let me check. He has a name for that system, but I have it somewhere in my notes.
Well, okay, it's probably in the paper. Okay.
Biotherapeutics. Yeah, let me pull up his. I think it's his. Yeah, there it is. It's in his link. I hope it's still working because he shared it. It's his Google Drive link. No, it's still the same. Oh, he didn't. Oh, he shared the link. I don't have his presentation. So yeah, if you want to learn more about it, the link for the presentation should be in the replay. So Dr. Risch, and then if you see microbiome, yeah, if you want to have the link to his presentation, know more about the ELP platform that he's creating that targets gut microbiota for prevention and treatment of human disease. Yeah, then go on his presentation, but it's ELBP platform. He named it that way. So
Okay.
engineered life biotherapeutic platform. That's where the ELBP comes from.
A lofty title.
At the at the Vis Institute. Exactly. Okay, and next we had Dr. Auti. I don't know. Were you here when Sam was here? Sam Otti.
I'm not sure what was the talk.
Let me pin the link really quick.
If it was in the evening, I probably was. Time? No, I missed the time crystals.
Yeah, yeah. It was a morning room. So yeah. So Dr. Sam Otti, he's at Lancaster University in the UK, and he's working mostly on superfluid helium and yeah, but basically superfluid discoveries is mostly what he worked on, and he has really cool questions in the bio. What does it feel like to touch a quantum fluid, and can we melt a time crystal, and whether the outcome is a time liquid? That was part of his bio. So that was really cool.
Oh, like a cup of quantum liquid. Huh?
Yeah, that probably not too healthy.
You know what happens if you drink it? Do you become more entangled?
Yeah, maybe live forever. Because the oscillations
Or
are
you're
so.
stuck in a loop?
Oh, right. Yeah, you're stuck in a loop. Oh, that's where you have, like the famous movie
Like
where the day always starts.
Groundhog Day?
Yeah, exactly. Drink a time crystal and you have to Groundhog Day. No, I'm kidding. It's total nonsense. Please don't listen to me. So, so he basically talked a lot about how the setup is to make a time crystal, and that it's kind of an impossible, thought to be impossible, because if you want, it's kind of a perpetual motion machine. And we had the time crystal room before, with the Google senior researcher, and the thing is that these time crystals go on. In theory, they would go on and on forever if they are completely isolated um, between those two oscillatory states. But they are not. They are never completely perfectly isolated. But he got them to in the state for 1 second, which is crazy. It's such a long time for quantum states. So, uh,
Okay, so that's a lot longer than the other
yeah.
time crystal.
And the thing is, why is he explained, is because they increased the number. So they have now 2 time crystals. And these 2 time crystals, they are basically in a non-linear to level dynamic. And yeah, he explained that because of that, they could increase the stability and also the time, which was really impressive, the time. And yeah, you can see here in the presentation the setup basically, where he has this different chambers and the helium shield, and also the NMR coils. The setup was explained pretty well, and then you have this 2-level system that basically increased the the time.
So it just keeps oscillating back and forth at some frequency between two states.
Yeah, but it doesn't create any work, right? So that's why it
Right.
works. Kind of not. Yeah.
Well, so did he say what frequency it oscillates?
I have to check because it's not in the.
Yeah, I'm not seeing. Oh, I see. It's no, that's the decay is over seconds.
He generated 10 or 12 magnons, The processor on the external that associated with the magnetization of processor around the external magnetic field. Oh, that's just the magnetic field. In the system, the observed time crystal was extended to up to 1,000 seconds, and the absence of the driving force.
Oh, well, on page 4D, it shows frequencies decaying. So they get slower?
Yes, until it then finally doesn't oscillate anymore.
I see.
But
So it bleeds off, but and then gets slower as thermal motion kind of knocks it,
yeah,
slows it down.
and
Okay.
it would also increase the temperature in this setup. It's not as the need to not as cold as it needed to be before in the study. We heard before, but
Mm-hmm.
I'm checking.
Oh, and the coupling does interesting things. Hmm. Well, that's kind of cool. So they don't know nobody's speculating what you can do with these things. They're still cool.
So coupling extracted from the sideband extrapolates to 1.7 plus minus zero point 0.4 hertz at the crossing in good agreement with the fitted simulation value. Yeah, okay.
Cool.
Okay. Yeah, the temperature. I'm trying to look up the temperature, but I can't seem to. Yeah, I don't. In the slides, there's not too much mentioned. I don't think we asked about the exact frequency, to be honest.
Will they give some chart? But yeah, it's weird that the frequency would decay. I guess it's not weird, but it just says what it's doing as things knock. I wonder. Oh, hi, Fasudev. I wonder if you have any comments on time crystals. But yeah, this was kind of cool. It's impressive it lasted a second, but I didn't know that the frequency decays as well. So it's not a fixed frequency oscillator. Huh.
Yeah, I think they started at a specific frequency, and yeah, but it decays over time. I'm just trying to figure out the resonance frequency of the tank circuit can be tuned in 8 equidistant steps between 550 kilohertz and 833, and corresponding to the external magnetic field.
Oh, I thought that what was the chart on 4? It looked like on 4E, it looked like it started at 20 hertz.
So it's 16.5 megatesla and 25 megatesla. Like it's the external magnetic field.
Was the
And
magnetic field oscillating in strength?
yes.
Oh, that's what was driving the oscillations of the time crystal.
Yeah, and I think to start it off, at
Well,
least.
isn't that putting work into the system then?
So no, I think they'll just I think the idea is to then leave it alone and isolate it, and then see if it keeps going for um, how long it keeps going on. And I think they got it to 1 second.
Okay, so they just pumped it with external fields and got a pitch, and then that decayed as the crystal decayed. Okay, cool.
Yeah. Okay. Yeah, it was cool to have that increase of time, and yeah.
Hmm. Okay.
Then we had Dr. Jill. Let me pull up the link. Dr. Gill. I'm sorry, it's Gill, not Dr. Gill. And here's the link. I'm not sure if you were there because I think it was a. Oh, but wasn't it Friday? I think you were there, right? It was Friday morning. The multi
Transient,
room.
yeah, the transient reprogramming. Now this is familiar. Why
Yeah.
is
So
this not?
yeah. So he did his PhD in Cambridge, UK. Um, and yeah, he is working on understanding how the epigenome changes as we age and how these changes are reversed during iPSC reprogramming, and particularly focus on transient reprogramming approaches, which promote rejuvenation while maintaining the original cell identity, and that's quite important. So he doesn't change the genome in any way; he changes the epigenome. So how the genes are being used. And it was really impressive that with his reprogramming, so he basically established a protocol of the ideal reprogramming type and time, um, to basically rejuvenate cells. And yeah, did a lot of um, transcriptional analysis, um, of different gene expression analysis, how they temporarily are being regulated, and what type of reprogramming changes that. And he also looked at collagen, which is really cool because when people used to say, it will be impossible to rejuvenate collagen. Um, so,
Yeah,
but
that's
apparently,
really cool.
yeah, it's not
Well, good.
impossible.
So he's just applying the what is it, Yamanaka factors, right? For programmed periods of time. So there's a gradual regression in the age of the cell through demethylation of the DNA. Is that basically what's going on?
Yeah, not just methylation, also histone, like how the genes are folded. Um, so it's not just methylation. Yeah, and so he activates this rejuvenation reprogramming with doxycycline. So and so basically there's a way to have um, this reprogramming happening only when a specific compound is present, and that's how you can time it. And he could basically he found like a sweet spot. If you do it too long, which was done before, it kind of messes things up again. So you have to kind of find the sweet spot to have rejuvenation and not mess up things then further along. Yeah, so he could turn back basically this epigenetic clock, and without changing basically the identity of the cell. So yeah, he didn't wait so long that the cell's identity would go away, like there would be a stem
Mhm.
cell or something like that,
See, I
which is
couldn't help, chemically you'd expect if there's increasing methylation in time, that's going to change the physiological properties of the chromosomes, and they're going to fold differently just because their interaction with water. And it kind of implies that, well, perhaps part of the developmental strategy is to position, is the reason that, or at least some component to aging is there's the gradual changing in the folding of the DNA, which will expose different uh, DNA or make them more readily available for transcription. And it's an interesting kind of strategy to sort of encode the expression of genes over time. If you're systematically methylating and driving a different change in the fold or the quaternary structure would be tertiary and quaternary, of the chromosomes that may expose in different periods of your lifetime, expose different genes for, kinetically for expression, which was kind of cool because that might imply some of the mechanism for how, large groups of genes will turn on or off, or if there's a critical transition in the fold that may facilitate, large changes in development like puberty or other, menopause or other, dramatic changes in the phenotype.
Yeah, exactly. So yeah, during early development, it's really crucial. That's how the cell basically changes, like grows into the fate they were, they're supposed to become. But then with age, it's basically kind of, yeah, with increasing age, things become unavailable, like methylation builds up and folding occur, that basically turn into like into bad proteins that then accumulate in the cell, and the cell can't keep up with it, with controlling. And then since also these, additionally to that, these non-coding ancestor viral DNA becomes also more active, since um, over time the cell control system has just more and more errors to repair. And as soon as you don't actively suppress those, they become active too. And then you basically have so much big of a mess in the cell that they start, age and dying. So, and this is basically repolishing a scratch DVD. I think David Sinclair used that at some point to explain it. So
Well,
yeah.
and I wonder if, as you're exposing these ancient viral DNAs, if that's an indication of, the, sort of average age where those infections, were there in the first place. If there's this, gradual refolding, then it could indicate that, that was the whatever they were doing in at that period of their life. That's when they were vulnerable to those infections.
Yeah, yeah, exactly. So whenever the cell is under really high stress, these viral DNAs, they become very active, and they make copies of themselves and duplicate themselves in the genome. And the more
Hmm.
this happens, the riskier it becomes for the cells. And then the theory is that, for example, disorders like ALS and neurodegenerative disorders, they are highly involved. These, that the cell is just not, for some reason, in that person able anymore to cope with it. And with ALS, especially, people think that because was a really interesting few cases that NIH and lab at NIH observed, that people that were on antiviral drugs through HIV treatment, a long-term treatment, um, they were like they had this genetic severe form of ALS, but it never broke out really. They were already in their 60s and they were still doing really fine. And that's how people had like the first indicator that this may be due to some viral activity in cells, and our ancient viral DNA. And yeah, so it's a really interesting field, the epigenome. It's really complicated because it adds so many more levels of complexity to the genome. And knowing all of it, it's basically impossible. I don't know. No, it's not impossible. It won't be impossible, but for now, it's really hard. So it's a really great study in the right direction to look at also the timing, not just what mechanism to activate to rejuvenate, but also look at the timing and then screen for what it's actually doing. So this will, yeah, this work will contribute to many more, not just aging, but also I think all kinds of disorders like chronic disorders that are uh, very common in the general population, like diabetes and things like that. I think it will also contribute to find treatments for those disorders, Parkinson and so on. So yeah, the early detection would be another thing. And we had a guest speaker here a while ago. I don't know if you remember. I think it's Dr. Belsky from Columbia University. Was that him? That came up with a fairly easy to use blood test where you can basically find. I'm so sorry. Where you can basically find in a regular blood test the aging clock that you currently are on, and that's also reversible. So basically, if you eat healthy, have less stress, and so on, you can see that this methylation aging clock kind of stops or it goes even back a little bit. So he really sees it from a public health perspective, where he wants to look at, stressful childhood, people that have undergo scarcity throughout their life. And he developed this blood test to screen basically how it affects them in the aspect of like chronic disease, aging, and it's really cool that people are working on this. So yeah, it can increase our quality of life by the end. I think we are just what we are doing right now is just extending life, but not life quality. Um, and I think this type of work will maybe, I don't know how much it will extend life, but it will for sure extend hopefully life quality of life in the end also. So I think that's really important.
Yep, very cool.
Yeah. So uh, let's go to our next one. I think you were there, right? The Friday physics um,
Oh yeah,
cosmology.
this was with the simulation with the the universal simulation.
Yes. Exactly.
That was a fun one. There
Yeah,
was some good discussion on that.
that was fun.
I
Mm,
want to render their data in a metaverse and fly around in it.
yeah, right. That would be so cool. And he had that YouTube video up. If you are interested in looking it up, yeah, if you go on to the recording, there you can have the YouTube video.
Yeah, it was a great video. Oh, go ahead and
Oh no,
describe
no, go ahead.
for the. Well, I mean, it laid out the. So it was interesting technique, and there was a lot of discussion about it. But they had to find the initial conditions that once they so they balanced the dark they injected the dark matter and the dark energy, and they had to balance those in a way with the observable matter such that when they ran their initial conditions forward, it would pass through the known data that we have. And they ran about 50 trajectories. Each took about a month on the Tokyo supercomputer, so this was a completely heroic simulation. But then they averaged the 50, and that was the data. What's interesting about what given that the constant velocity of light, the farther over the simulation volume um, that we're looking, the farther back in time. And so they it was an interesting projection over the up to the horizon of what we can observe, which would go back to 14 billion years. But the data had to, in essence, pass through that point where it explained our observation uh, window out to our horizon. But then what's cool is it had, the simulation contained, the rest of the universe as it would be today, the predictions of the structure. And that informed some of the, that would make predictions of certain structures that would form that we're not able to observe yet, but they're already, you know, present today, but the light from them just hasn't arrived on our telescopes yet. So, really cool. They were able to make some, indications to the folks that are, have access to the Webb telescope as to what kinds of structures and in what directions, there we might see early events of. Um, so that's really kind of cool.
Yeah, yeah, I really like the discussion and the room, and also, yeah, the discussion we had afterwards, how the universe is expanding, how at some point we will basically lose contact to galaxies, and the network will basically lose contact. What does it mean on the quantum level? How things will change, we don't know. But it was interesting to discuss about it, and um, also, yeah, about his basically time machine to see how things evolve. Possibly, we cannot 100% know because he explained how in the beginning we have this set conditions that we are relatively sure about, but the further along time goes, the more options we basically have and the more unknowns. And this is basically one model, but it's fitting quite well, I believe, um, as far as we know. And it came up with these structures that remind me of the cytoskeleton for
Yes, cytoskeleton, astrocytes, fungal networks. It was interesting to see that the whole web structure, how it looks similar on so many different length scales.
Yeah, it's unbelievable. And the scale, and this is just the observed universe, it's not even the
Mhm.
whole universe.
I
So,
thought there was an interesting set of side questions about in entanglements, and I got off on a tangent about um, the for photons that have gone beyond the horizon, that well, there can't be any communication with that part of the universe again, that happen to be entangled, and we measure their spin. In essence, we start to know things about the universe that that is no longer connected, which is
yeah,
just kind of cute.
it
Because
is. Oh, go ahead. Sorry.
no, I mean, well, but yeah, I didn't get around to formulating a question about that, but there was some discussion about um, you know whether that's what that would mean.
Yeah, yeah, I asked this if you can imagine like an evolution, the Galapagos Darwin um, birds, when they are isolated, they kind of, if they evolve in a different way and if kind of the rules could change after, if they have kind of a different evolution, basically, because they are not connected to us, like, to the whole network anymore. So, what will happen to it um, over time?
Yeah, just to the fact that increasing amounts of matter dropping off the horizon, does that affect our dynamics?
Yeah.
And it's kind of cool.
Well, the question is, will it kind of completely split off? Will that become the multi-universe? I don't know. Um,
Yeah,
like a
that's the
cell
multi,
that is,
all these universes beyond the horizon,
yeah,
or
like a
parts of the
cell.
universe.
I don't know. It's interesting to think about. Then the fantasy takes over, and it's not fact based anymore in my head. Uh, yeah, it was really fun to think about these things and to have this discussion. It was amazing.
Hmm.
And that was our week. Uh, I think it was pretty cool. And so we had already a room today, earlier today, and that was interestingly also about galaxies that are far, far away and looks back in time, basically. Well, they are candidates for galaxies. We don't know yet. So that was, but we'll discuss more in the next weekly recap this room. But it was interesting because um, they are very far, the furthest away observed galaxies, and they're candidates for galaxies, or they are maybe these primordial um, black holes. Do you remember we had the primordial black hole that generated all the dark matter energy room that we had? Were you there?
I don't think I think I may have missed that one too.
Oh,
That
well,
was a theory that the dark matter came from. Oh, well, although it sounds familiar. So there was a lot of little ones, or it was a massive one that.
yeah. So these primordial um, black holes were really massive, and they occurred basically in the first second of the Big Bang in this very massive state, and they the that was basically a model paper that discussed that this is basically the scenario, the ideal conditions to generate all this dark matter energy, because we don't have these conditions that make any sense mathematically anymore. But under those conditions, the model fitted really well. But we are not sure if these primordial black holes actually existed. And so the talk today was about this very, very far away galaxies that they have weird properties. So they could also be maybe a candidate for these primordial black holes. We don't know 100% yet. They just described the characteristics and what it could be, and they will collect more data. And then I asked if looking at it with the radio frequency would help, and he actually said yes, because black holes um, generate you can detect them if they are black holes in a very wide spectrum of frequencies, um, and actually that you could basically, if they are black holes, you could also detect them with radio frequency detection. So yeah, he said, oh, maybe we could collaborate with this and this that has this detection, this biggest detection device. So
The
yeah,
radio telescope.
yeah, the biggest one. We would need the biggest one. So yeah, I hope he said he would come back in the fall or winter. Maybe he has some updates about.
Oh, cool. Were these quasars or something? I don't know, because quasars, were they quasars?
No,
Oh, okay.
they were this age one, and he oh, let me go in and get the paper. I can just link it while we are here, anyways, because it was so interesting. And he was really good at explaining. starburst galaxies or quasars. They don't know what they could be, or
Oh,
they.
so they could be quasars.
So
Okay.
yeah, they are. But if you look at it, they describe them um, with very distinct properties that are not common in galaxies we see, like in closer by, so newer galaxies. Um, so it could also be, black hole growth in the sources are quasars. Could be. So yeah,
Hmm.
it's really interesting.
Cool.
Yeah, so here they just describe their physical nature, basically based on um, if they would be galaxies that are forming stars. They are creating stars at a very, very high, very unusual and very high rate, which is very surprising. So they would be extreme star formers, which is it's just a cool
Oh,
word.
so and that's where they think um, a lot of dark matter was involved in the
Yeah.
increased rate of star formation.
Yeah, so yeah, exactly. So this observed luminosity that could either mean there are a lot of stars formed at a very high rate, or could be also black hole, and that would require heavy seeds. So yeah, but for that they need to look at it in more ways to get a more accurate, uh, more accurate picture.
Mm-hmm. That's
Interesting.
cool.
Looking back in time again. So yeah, we'll have a room tomorrow morning, actually at 10 a.m., where Dr. De La Fere, she will look at. She made an AI that consists of neural nets, and the neurons have dendrites, um, and
Oh,
really
she
cool
she
dendrites.
increased the model. Oh, that's cool. I
And
want to see how
that
she did.
increased. To increase the stimuli detection with creating basically this neural net with dendritic neural net. So that will be the talk tomorrow at 10 a.m. And
That's
then we,
really cool. I mean, I'll have to catch the replay because I can't make
yeah,
it. But okay, the
I'm sorry,
replay will be.
it's from,
Well,
yeah,
that's okay because then I want to look over the paper too because I really think we need. Well, in order to even get to modeling the astrocytes, we need to better models. We need a dendritic model with synapses
yeah,
to even form the tripartite synapses. So
yeah, it will be
cool.
interesting to listen to her. And then we'll have on Wednesday at 9 p.m. Dr. Armbruster. And this is a room just for you, sorry. New function astrocytes discovered in the brain.
Oh,
So,
it's in the evening, I hope,
yes, it's at 9 p.m. Yeah.
and that's Wednesday,
Yes.
and it's even got astrocytes in the title.
Yes, it does.
Cool. I'm definitely gonna make it.
And then we'll have on Thursday morning, 9 a.m., Dr. Kiehi, I hope I'm saying the name right, talking about self-assembled logic circuits made from proteins, which will also be interesting.
From proteins, huh?
Yeah.
Okay.
And then Dr. Kihu, it's on Thursday evening, like at 9 p.m. EST, talking about the expansion of desert climate in Central Asia. So it's a climate room. And on Friday at 1:30 p.m., we'll have Dr. Kagan talking about gene mutations across species and how they shed light on aging. So yeah, another aging room on a Friday.
Cool.
Yep. So that will be our week, and yeah, thank you for coming, everyone, and thanks, Serena, for being here.
Mm,
And, yeah, enjoy the rest of your evening, everyone, and or morning or wherever you are. And
okay, thanks everyone.
bye.
Bye.